Hello everyone, today I'm going to present TAIL, transformer-based Protein function annotation with joint sequence label embedding. So understanding protein sequence function relationship can provide enormous benefits for biological and pharmaceutical applications. However, significant gap exists between the availability of sequences against the availability of their functions. Therefore, we introduce a normal transformer-based deep learning model, transformer-based protein function annotation with joint sequence label embedding, also called TAIL. So our results demonstrate that TAIL Plus outperformed all sequence-based methods in all three ontologies and outperformed the state-of-art sequence plus network method NetGo in PPO and CCO. And even when network information is not available, TAIL Plus outperformed all compared methods in all ontologies. And moreover, we have shown TAIL Plus showed superior generalizability to sequences of low similarity, never annotated new species, as well as rarely annotated functions. So here is a summary of our method. So first, TAIL actually replaces the previous used CNN with self-attention based transformers. In this way, TAIL can capture long-term dependencies better in the protein sequences. Secondly, TAIL embeds the sequence inputs and the hierarchical function labels together into a joint latent space so that our model can deal with the tail label situations. And thirdly, we actually consider the hierarchy among labels through regularization during training, unlike previous method, which consider this in the post-training way. And finally, we propose TAIL Plus by using an ensemble of top tail models and a sequence-based method Diamond. This idea was inspired from DeepGo Plus. So here is our model architecture. So basically, our model has four major components. So first, the sequence encoder, the label embedding, and the output of the two will go into a joint sequence label embedding, and the output of the joint sequence label embedding will go back to reweight the output of the sequence encoder, and eventually going into a FC fully connected layer and the output layer. So besides the model, we actually introduce a new loss functions, which is the sum of a cross entropy term, L plus, and a novel hierarchical regularization R here. So this hierarchical regularization is to mitigate the hierarchical violation where the predicted score of a child geoterm may be larger than those of the ancestors. So finally, we actually use an ensemble of the tail and plus a sequence-based method, diamond score. And we use a convex combination of three tail models and the diamond score together here. So for the results part, we actually first compare tail plus and all its competitors on the kappa 3 test set, our test set, and the portion of our test set without network information. So as you can see here, on the kappa 3 dataset, TAIL Plus actually reached the best on BPO CCO based on Fmax and UPRC against all its competitors. And on MFO, TAIL Plus actually reached the second best next to GeoLabeler. And on our dataset, actually you can see here, we can reach the similar conclusions. And finally, to the portion of our dataset without network info, we can see TAIL Plus actually reached a state of art compared to all its competitors, and in all three ontologies. So secondly, we will show the generalizability analysis. So specifically, we'll show the generalizability analysis to the proteins of new species, low similarity, and rarely annotated functions. So first, on the new species, we can see here, we compare TAIL and all its competitors, including Naive, Diamond, DeepGo CN, DeepGo Plus, and on all of these six spins. So these six spins are actually um, the, the proteins in the test set on three ontologies and on eukaryotes and prokaryotes with new species compared to the training set. So as you can see here, based on the Fmax, TAIL and TAIL Plus actually reached uh, superior performance compared to all its competitors on all the six spins. Then on low similarity situation, we actually calculate the maximum sequence identity of each test sequence against the training set. And we split them based on this MSI into 4 bins. And you can see here, tail plus and tail actually on those low similarity region has the best performance against all its competitors. And finally, on the real annotated functions, we actually also split the test set into 4 bins based on the average label frequency. So as you can see here, on the low label similarity region against the training set, tail and tail plus actually reach the best performance on the most of the cases across 3 ontologies. And that's all. Thank you.